بكم جميعا. انا عايز ارحب بكم النهارده في الميتنج بتاع النهارده. يعني حقيقه ريل بليجر وشرف وونر تو ميت يو اور فيرتشوالي. انا شايف حقيقه بانل فيري ديستنجوشد، اساتذتنا ومعلمينا من زمان، يعني اساتذتنا. مبسوط جدا ان انا شايفه النهارده فيرتشوالي. حقيقه برضو ده شرف كبير ان الميتنج ده يبقى برعايه الجمعيه بتاعتنا، جمعيه الاي اس ام تي. مش بس بتابجريد الاتش بي براكتس في مصر، بس ليها بارتيسيبيشن هايل في الكوميونتي، ونعرف الجمعيه من زمان، وفعلا حاجه تشرف وحاجه فخر للدكاتره المصريين. فانا فعلا يعني مشرف النهارده ان الميتنج ده تحت رعايه الجمعيه، خاصه انه احنا في ساندوز قررنا ان احنا مش هنستنى تو لونش اور ترانسبلانتيشن بورتفوليو لغايه ما الازمه اللي احنا بنعدي بيها دي ككوفيد 19 تعدي. قلنا خساره الوقت ده، لازم نوصل على قد ما نقدر بدري لدكاترتنا والعيانين بتوعنا، خاصه ان احنا عندنا فيري هاي كواليتي ميديكيشنز هتساعد عندنا في مصر. الحقيقه طبعا زي ما حضراتكم شايفين الظروف، احنا بنتقابل فيرتشوالي بس، والعلم الحمد لله بيلاقي الطرق بتاعته عشان نوصل علم اساتذتنا لبقيه الاس سي بيز كلهم. لكن على وعد ان شاء الله نتقابل فيس تو فيس اول لما البانديميك او السيتويشن يتحسن، يا اما نرجع للنورمال لايف بتاعتنا ان شاء الله سون باذن الله، او النيو نورمال، هنشوف الدنيا هتبقى عامله ازاي. بس كلنا امل ان شاء الله نرجع نشوف بعض فيس تو فيس، لانه طبعا مقابله حضراتكم فيس تو فيس غير يعني. فان شاء الله على امل قريب نتقابل كلنا. انا الحقيقه النهارده برضو بتشرف ان دكتور هاني حافظ هو اللي بيرأس الميتنج، وانا مش عايز اخر اكتر من كده. انا ادعو دكتور هاني حافظ بروفيسور اوف نيفرولوجي كايرو يونيفرسيتي والبريزيدنت بتاع الجمعيه اي اس ام تي بتاعتنا to lead and open the meeting. Professor Hani, over to you يا باشا.
Thank you very much, Dr. Sameh. It's a real pleasure and honor for me to be with you today and to see you after a gap of absence. And you are coming as a GM for Sandoz. It's a great pleasure for all of us. Of course, transplantation in Egypt dates back to 1976, when in Mansoura and in Cairo in the same year it started. And 3 years after that, the Egyptian Society of Nephrology and Transplantation started. So actually the ESNT in Egypt is almost the same history of the transplantation in Egypt. And we have the privilege of being a unified society all over these years, in spite of other branches who have been divided into many societies. I don't want to mention the specific, but let's see the gastroenterology or the endocrinology. You will find many societies dealing in the field, which is not the case in nephrology. Also, I am proud that we have now a higher committee of transplantation, giving the license for all the transplants to be ethical in Egypt. And we have a law that has been now since 2011. Unfortunately, we didn't reach yet the cadaveric program because of some logistic steps. But we are hoping that in the near future, after the era of COVID, we go either to paired donation or to starting the cadaveric transplantation. And I am honored today to have co-chairs Professor May Hasaballah, the president-elect of the Egyptian Society of Nephrology and Transplantation, and my dear colleague in the Cairo University Faculty of Medicine, since we were not only professors, but when we were students also. And also Professor Adel Bakr, a dear friend since many, many years, and a colleague in all the pathway of transplantation over, I don't want to make him old, but it's almost about 35 to 38 years working together in the field of transplantation. I will start by presenting Professor Adel Bakr, who will dip in the history of transplantation in Egypt and will give us much more details that will be enlightening all the audience. Professor Adel.
Okay. First of all, I would like to thank Sandoz for kind organization of this meeting, especially Dr. Sameh. And I would like to thank to my dear colleague, Professor Hani, and dear sister, Professor May, for chairing this important session. Moreover, it will be my pleasure to present the history of kidney transplantation in Egypt on behalf of the old Egyptian nephrologist. Okay, I will start by short background, then discuss with you in some details the history before reaching the final part of my talk. We are here today because we have a good history yesterday of renal transplantation. I would like to make a flashback for the history of transplantation worldwide. In 1952, Hamburg J, the first living transplantation, mother to son, in Paris, France. Kidney was functioning for 3 weeks, then rejection, and this because he had no immune suppression at that time. While first successful identical twin transplant were by Joseph Murray from Harvard, Boston, USA. In 1962, first unrelated transplant with total body irradiation and steroids and 6 mercaptopurine, while first cadaver heart-beating donor was done in Belgium in the same year. This is the first report for history of renal transplantation in Egypt. It was in beginning of 2000, as it is an invitation from Tissue and Organ Journal in Italy. At that time, we had 20 transplant centers performing around 5,000 transplants. And at the end of the same year, Professor Barsoum and myself published a paper around clinical transplant book by Michael Cheka and Paul Torazaki for Egyptian renal transplantation. As regarding the history, it is my pleasure that Professor Barsoum kindly accept my invitation to write the chapter of Egypt in my book, 2017. And according to his system, it is a systematic approach, organizing a questionnaire, then select questionnaire participants and collect the feedback from the doctors. The questionnaire including, is there a nephrology practice in your institute? Yes or no? What about the dialysis, first dialysis in your center? What about the transplantation activity? What about separate specialty of nephrology in the center? What's the educational entity for young nephrologists and research area for the young generations? The questionnaire participants distributed all over Egypt from here and there. Continue the list. Besides that, further specific data were added by Ahmed Adam, Sabur, Zanati, Al-Attar, Zaki, Fayyad, and Ashraf Dunya, while Professor Barsoum was so kind to provide us with data of Cairo University and his center. What about the feedback, collecting the data? Unfortunately, first transplant tourism was carried in Medical Research Institute Alexandria 64, where the patient was Madame Julia, a Greek woman. The donor was an Egyptian. The surgeon was an American, Dr. Kuhn. And unfortunately, again, the assistant was Professor Abu El-Ainin, not Hassan Abu El-Ainin from Mansoura. The first 2 renal transplants, as Professor Hani said, were in Mansoura. This is the upper picture is Russia, mother of her daughter, Ra'ia, the first donor and recipient in March 76, while a second transplant were in August 76 before Cairo present first transplantation, as I will say later on in October. After major surgery for Ra'ia, has middle line incision with bilateral nephroerectomy and right iliac renal transplantation. After initial period for diuresis for 12 hours, the patient went in ATN. Supported by dialysis, and after that start to produce urine and creatinine went down within 2 weeks to the normal, and the patient did well for the next 20 years. And Professor Ghunem announced this in the national meeting of Egyptian neurologists 78. Mansour team as a surgeon, Professor Ghunem, Professor Hamadi, we lost him last year, Ashamallah, Gaballah, and Bayoumi. Nephrologist, myself, Nabil Hassan, Muhammad Sobh. I established the first 2 clinics for preparation and post-transplant follow-up in 79. I'm responsible for creation, AD, shunt, and fistula, and training both nephrologists and urologists for that. Established 3 hemodialysis units, the main hospital, and 2 satellites. Organizing morning transplant meeting, discussing emergencies and new admissions, besides being an administrative chairman before being a chairman of the unit. Nabil Hassan followed me after completing military service. Sobh followed after 2 years when returned from Canada. We performed 43 transplants in 6 years. Then we shifted our activity to our urology nephrology center 83 with celebration of 3,000 cases with contribution of many generations. Some data about our experience from March 76 to March 2016, 40 years experience exceeding 2,700 transplants. Okay. This is the overall actuarial survival, patient and graft for 20 years. This is significant factors by univariate analysis, revealing some factors: donor age and recipient, number of blood transfusion, original kidney disease. But adding Cox proportional hazard analysis to have the multivariate that revealed only 5 factors sustained their impact on the transplantation. The first one is the donor age. As you see, the best result for 5 and 10-year graft were younger age, and also the best graft half-life was with this age. Second, genetic consideration according to Gerhard Opelt in CTS. HLA identical sibling is the best 5 years, 10 years, and even graft half-life, while 1 haplotype mismatched related and 2 haplotype mismatched either related and related follow the result. As regards the immune suppression, coming to the top, tacrolimus-based therapy, 5 years, 92, 10 years, 86, half-life is 21 years. Rejection, no rejection, or 1 rejection, or repeated rejection. Of course, no rejection is the best. Steroid dose in the first 3 months. When you use less, have a better result for 5 and 10 years and the graft of life. The most important part is this equation as collaboration with Cleveland Clinic. SCT equals SOT expanded Y, where SCT is the probability of survival and SOT is probability of baseline survival, and y is algebraic sum. So we have estimated range from 0 to 2.6. This is further classified to categories from 0 to 0.6, 0.6, 1.3, 1.3 to 1.9, and 1.9 to 2.6. Having the baseline function of 96. What is the baseline function? Is the best parameter in each variable. Say you have no rejection, the chromosomal immune solution, so and so. So the graft to survival at 5 years is 96. Cairo team, first transplant, as Dr. Hafiz said, 76 October. Two brothers from Dr. Barsoom's clinic. Surgeon were Muhammad Safwat, Hazim Turk, Abdul Qadir Ut, nephrologist Professor Abdinam, Professor Rashad, and their sister Samia Buraya. Immunologist is their friend Dr. Mu'mina Kamil. Dr. Ghunem was invited and attended the operation as an observer. This is the CTS of OPEL. Lazar University started 2009, had 50 cases. Alexandria started 1990 and have 6 centers with total of 1,200. According to Magda Sharawi, total of Hanshams was 440. In Asyut, Tuhami started 89 with 25 cases. Nasr City with Sabri Gohar, 93, 2,000 cases. And Nasr Institute was both Professors Ramzi and Assam that we unfortunately lost them last year with around 1,000 cases. Arab contractor by Professor Abdul Menam Hasaballah, 550. Dr. Barsoom Salam and CKC, around 700. Military hospitals, according to Khalid Aweda, there are 10 hospitals, started 73, and have cases either from army or from general population. And Khalid had 6,000 cases, out of them 13 were kidney, liver transplant. Misr International, according to Muhammad Nasrallah, 3,200. Zaid with Salah and Sisi, 700. Sohag, 20 cases. Zagazig, 9 cases. Nirbadrawi, I tried to take the data from Sahir, but started 86. Azhar, Ahmed Maher, 4 cases. And with Walid Masoud, have 14 cases in Toshiba. Urology Nephrology Center has a transplant program with Abdul Ibrahim Abul Futuh, 88, and they changed their name. The leaders were Amr Ayad, Dunya, Fakhr, and Hassan, and others. They prepared for silver jewel with transplantation 1,000. First unrelated case was in Cairo University, who nursed me to a doctor. They discussed it in ethical way and transplanted it and paved the way for the unrelated job. We had first disease donor in Cairo when Attorney General approved harvesting from executed criminals and transplanted in by Midhat Khafagi in Cancer Institute, but had a big debate. Then we have a law, as Dr. Hani said, 2010, not yet activated, and now we have MOH registry. And if you allow me to conclude, Mr. Chairman, as you said, Egyptian site of transplantation was founded by Professor Hasaballah 93 and became dormant after death of Professor Ghunaimi and revived again by Abdulhamid Abaza, and now it is a fundamental leader in transplantation. Okay, and the Arab Society I was a vice president for 4 years. I am one of the founders of the Method 88 and was also a WHO consultant in nephrology to Libya, a member of Istanbul Declaration Steering Committee and Summit, and organized the fifth meeting Istanbul Declaration Custodian Group in Cairo, March 2010. And one of the members, I was one of the members of the KD guidelines in living donor transplantation. Lastly, the total centers now was doubled, around 40, with total transplant in Egypt, according to MOH registry, exceeding 20,000. Out of them, 1,300 in 2018 and 1,400 in 2019, and till now, only 320 as the continuation of the problem. And in my book, Professor Wanem wrote the introduction to assess or to suggest Arab transplant to supervise the distribution of cadaveric organs, to set up a regional database, to raise the funds, and to support the programs in less privileged centers, besides supporting research activity and organizing a meeting. Please, I do apologize if I miss some centers or had a mistake in presenting any data. Please contact me to correct in this email. Fortunately enough, this is the most recent publication in the Transplantation Journal, where I received the invitation from Stephen Toulouse, the editor-in-chief of the Transplantation Journal, to write renal transplantation in Mansoura, Egypt, with this map, with our experience. And lastly, thank you, Sandoz, for organization, and Dr. May, Dr. Hani, for chairing this session, and thank you. Okay, Dr. Hani.
Thank you very much, Dr. Adil. Fantastic presentation. Thank you very, very much.
Thank you.
And also thank you for the book, which is a good reference for all the generations.
It is my pleasure, dear Hani.
It's my pleasure to introduce Professor Magdi Sharawi. He is a professor of internal medicine and nephrology at Ain Shams University. He is also a member of the board of the Egyptian Society of Nephrology, and he will be talking about improving outcome in renal transplantation. تفضل تمام.
بسم الله الرحمن الرحيم. أنا الأول بشكر ساندوز على دعوتي للقاء الجميل دوت، وفرصة أقابل أساتذتي وزملائي الأستاذ الدكتور عادل بكر والأستاذ الدكتور هاني حافظ، وطبعا أستاذتي الدكتورة مي. شكرا جزيلا على التقديم. It's my pleasure to be with you today with Sandoz meeting. My presentation today will be about improving outcome in renal transplantation. I think we all know, all those who work on transplantation know that this is the best option for a CKD patient is to be transplanted. Survival benefit all over the age group, and even in diabetic patients, they have a higher survival rate. And although the survival is very good in these patients, however, it doesn't reach the survival of the normal population. So there is still a room for improvement in outcome in transplantation. And this improvement room include many topics, as you see in my agenda. I'll speak about them all in sequence. First, how to improve donors and community education, because this impacts the outcome of the dialysis. Patients should know that they have a better life, a better survival time if they are transplanted than continuing on hemodialysis or peritoneal dialysis or even daily dialysis. And this survival benefit is best when the patient is young age and not old age. Even in high risk group, in patient who has incompatible transplantation, still they have the benefit of better survival than those who stay on dialysis. And this benefit include all aspects of life: physical activity, pain, mental health, emotion, function, vitality, general health, and also with less comorbidity like heart diseases, cardiac event, ischemic heart disease, MI, and any hospitalization, it is better in transplant than in dialysis patient. Also, the community should know that the donation is not that harmful. Of course, there is some minimal side effect of donation, but the average life that can be lost by donation by this very interesting study is almost 0.5 to 1 year for those who donate either male or female. So there is a little danger in transplantation that can be prevented if we selected the donor well. This is the threshold of who should donate, and if we respect this threshold and we don't expand the donor especially live donor criteria, we will get a better outcome. Also, the community should know that transplantation will save money for the community, which is very important to encourage transplantation. Other aspect is how to select patient for transplantation. We know usually this is a negative selection. We select the patient who come to us for transplantation, but we don't select the patient who actually need transplantation, like young patients, especially if they have mothers or fathers willing to donate. Patients usually don't hear about transplantation. They are depressed at the start of dialysis, and then they surrender to dialysis. There is lack of knowledge. There's a lack of training in the treating doctor and nurses, and also the fear of expenses and availability of donors from their family or others. So we have to encourage them to go transplantation. Also, we have to risk classify this patient in order to select the best immune treatment for this patient. And we have a lot of classification to risk stratify this patient. And there are some new risk stratifying methods we should use to select the best immunosuppressive drug. And this have proved to improve the outcome in transplantation. Again, we need to remember that better matching is good for the patient, especially if the donor is elderly, because elderly patients with many mismatches have the worst outcome in living transplantation. One other important issue that we should consider, especially here in Egypt, is the role of epitopes and ablet in selection of donors, and it affects the survival of the graft, especially in high-risk population. A very important aspect is we have to check patient compliance before transplanting them, because as we all know that the most common cause of failure of the graft is non-compliance. And we have to train the patient, to train the physicians, the nurses to detect this patient, especially those who don't have access to medical care, those who have longer time on transplantation, they spend a long time on transplantation, so they loosen up. And if the patient have non-adherence in dialysis or low health literacy, that's very common in this population. Another aspect of improving outcome is the good selection of the induction way. We know that nowadays most patients are using induction in the immunosuppressive protocol, and this became almost 80% of our patients using induction. And the long-term graft survival is best, especially if we selected steroid-free or CNI minimization protocols. And nowadays, almost 80% of our patients are using T-cell depleting agents like ATG, and this using the low-dose ATG have proven to have the same effect on prevention of acute rejection with good outcome and less toxicity of the ATG. Also, we have to, there is almatizumab. We have a little experience with almatizumab, especially in Egypt, because it's not present, but it is a very good induction protocol, especially in highly sensitized patient. And this abstract presented this year in the ATC showing a very good effect using almatizumab subcutaneously injected in the patient. It's much better than basilixumab, and it also reduces the need for a high level of immune suppressive medicated calcineurin inhibitors. And many patients also can use less mycophenolic acid or MMF in this induction protocol. But we have to remember that it is not free of side effect. It is associated with increased de novo antibody-mediated rejection, and also this year there is a publication of higher incidence of fungal infection in repeated introduction of almatizumab. A very new tried protocol is the low-dose rituximab with ATG to use it in steroid-free protocol. It showed a very good response. This is a 1,100 patient tried this protocol with good success. Again, to improve the outcome, we have also to stress on maintenance immunosuppressive medication. The evolution of these drugs, as we all know, started with the cyclosporin and then the tacrolimus, and then the clizumab, basilixumab, sirolimus, and everolimus until 2010. And I will show you some other new drugs. And these have changed the outcome of survival of the grafts markedly. Mainly, we are using now steroid calcineurin inhibitor, either cyclosporin or tacrolimus, mainly tacrolimus nowadays, and antiprotective mainly the MMF and mycophenolate mofetil. And some of us are using everolimus and sirolimus as an mTOR inhibitor. But we have to select better protocols for the patient, especially steroid withdrawal, and this will improve the long-term outcome and will minimize the comorbidity and the complication of transplantation. There are also CNI minimization or avoidance and other protocol. We will come through it. Steroid withdrawal, it is not, it's well known since long time because it is the most feared drug from the patients. But we have to remember, if we are going to stop or taper steroid, we should do it early, less than 3 months, with good maintenance. The best is CNI and MPA, with good induction, thymoglobulin or other biologic. Because the withdrawal of steroid after a long time after transplantation always carries the risk of increased rejection. But early withdrawal, like in many studies now, and in Mansoura University, they have a lot of steroid-free protocol, and in Shams also we have a steroid-free protocol. It has a very good result if we do it as early as possible, up to 1 week. Again, we have to remember that the best protocol to be used with steroid withdrawal is tacrolimus, MMF, and not other medication. This is according to this study. What about CNI minimization? We also know that CNI is associated with some complication, interstitial fibrosis, sometimes diabetes. And according to the Elite Symphony study, we now know that low-dose cyclosporin or tacrolimus are as effective as high-dose. So the recent protocol include low-dose tacrolimus and low-dose cyclosporin with MMF and steroid according to this study. And this is the best outcome. It was shown the best outcome in this study. The low incidence of biopsy-proven acute rejection is with low-dose tacrolimus and MMF. Of course, since 2007, there is a lot of studies about mTOR conversion from calcium, because the better effect on GFR and less CMV infection and less malignancies. But we have to remember that we have to do it as early as possible with good GFR and with the patient who has normal urinary protein excretion, and to avoid conversion in the late time. Also from Transform study, we have learned that combining everolimus with CNI, either tacrolimus or cyclosporin, is as effective as using calcium inhibitor with mycophenolic acid or MMF, regarding the outcomes, the GFR, and also with less CMV infection. This is also in this Athena 12-month study showing the same result. Combining TAC with MPA is equivalent to everolimus and TAC in this patient. But the most commonly used protocol worldwide, this is the OPTN SRTR annual data report just published, showing that more than 70 patients worldwide are using TAC, MMF, and steroid, or TAC and MMF. And we have steroid-free protocol in almost 40% of our patients nowadays, and this is the best way we know up till now. There are many other drugs in the, present for in the pipeline, but some of them are already used, especially outside Egypt, like belatacept. We know the benefit and benefit extension studies showing good effect. They use the co-stimulatory blockade to address the immune suppression, and abatacept also is there. And this belatacept have shown very comparable result with cyclosporin in GFR and outcome. With it was generally safe, with no increase in overall rate of serious adverse event or serious infection, and also it is less hyperglycemic. Less patient develop diabetes with belatacept, but it is not present in Egypt. Finally, we need to improve the outcome of transplantation by improving the long-term complication and to prevent rejection. And this means that we have not only to start with proper patient selection and the proper induction and maintenance immune suppression, but we have to detect T-cell mediated rejection as well as antibody rejection and the presence of de novo DSA. We have to monitor the patient. We have to monitor the metabolic and infection and the tumor state of the patient. We have to prevent if we can, and we have to prevent recurrence of the de novo treatment. And this is a very cumbersome function of us. But what we can do is we can prevent rejection, or we can try to early detect, especially antibody-mediated rejection, because we now believe that it is that much present in our patient. And biopsy alone cannot define antibody rejection exactly in all patients. From this study, from 46 patients with antibody rejection, the clear statement from the pathological report, only 18% we can be sure of them. And this is better with T-cell mediated rejection. So we need better way to identify antibody rejection. That's why we have nowadays, and we attended the ATC, there are a lot of paper on molecular microscopy that can define and can detect early the antibody mediated rejection. And we can use the molecular microscope to detect biomarkers, not only for rejection, but for drug targets. And this can easily identify patient at risk, and we can prevent early as well as late acute rejection in this patient, and de novo DSA, which will result in loss of the graft after some 10 to 20 years. There are many ways to detect this. There are many trials in the pipeline. There are trials using 3-gene signature, the key sort, the chemokine CXCL9 protein, CLCX10 protein, and also the donor-derived DNA, which is a very promising way to detect antibody rejection. The donor-derived cell-free DNA, again, it was presented this year and last year in the ATC, showing a very good possibility to detect it early. Also, we can use microRNA 15B and 106A and 374A to detect antibody and to prevent it as early as possible. Still, we treat antibody rejection by the traditional pathways, plasmapheresis, sometime ATG, rituximab, or IVIG. But there are several trials currently being conducted to prevent and to early treat late antibody-mediated rejection as well as acute antibody-mediated rejection. Some of them using the co-stimulatory blockade like belatacept, anti-PLIS, belizumab, anti-interleukin-6 tocilizumab. It's very famous now, tocilizumab, because of its use in the COVID-19 treatment. The proteasome inhibitor, bortezomib, the CD38 antibody, daratumumab, and the anti-C1 antibody membrane filtration, anti-C5 eculizumab. It is also tried. And also the IDs, the IgG degrading enzyme. We heard a very nice presentation by Professor Hani Hafiz just a few weeks ago about this new intervention to prevent antibody-mediated rejection. And if we review the literature, there are ongoing studies on eculizumab in IVIG, in bortezomib, calakitizumab, which is an anti-interleukin-6 antibody, bortezomib again, corticotropin, and mesenchymal stem cell. So the future carries many promising intervention pathways to prevent antibody-mediated rejection. Finally, if we can induce tolerance to this patient, it will prevent the excess use of drugs. And again, in this year, in the ATC meeting, there are they have this very interesting intervention pathway, the Mercury study, where they induced mixed hematopoietic chimerism and donor-specific immune tolerance, and then they will stop using immunosuppressive drug in this patient. Also, they tried exosomes in animal model to induce tolerance in this patient, and I think this will be the best way to improve outcome in renal transplantation. Finally, it is not one aspect. We have to improve all aspects to get an improvement in outcome. To my mind, the best recruiting patient is by education for them and for the community, and to encourage young patients on dialysis to be transplanted early, not to wait long time on dialysis, which will result in many complications that can be prevented and treated by early transplanting the patient. We have to have very good patient selection and stratification way to identify the best immunosuppressive protocol. We have to use all our tools in induction and maintenance. And as I mentioned, the best maintenance up till now is tacrolimus, MMF, and either with or without steroid. If we can prevent and detect antibodies and DSAs early, it will be very good. And thank you very much for your attention. Dr. May.
Thank you very much for this informative presentations. It's really a badly needed. We all, as transplanters, we want to get the best options for those patients to prolong the graft survival. So this is our main aim. Thank you very much.
Thank you.
Thank you very much. I think we can open the discussion now between the speakers. I don't know if Professor Adel Bakr is with us also. Professor Magdi is here, Professor May. And I think we can start until Professor Adel Bakr is with us with a few questions to Professor Magdi. You mentioned, Professor Magdi, that alemtuzumab is gaining a new concern recently. But you remember that most of the studies, the previous study of alemtuzumab in the past, were not very good in outcome, with very resistant type of rejection. So what do you think? We still need to try it, or it's better to go for the protocol that you mentioned of using maybe a combination of rituximab in small dose and the ATG, for example, which are more, has been applied in many patients with better results. What's your opinion?
Professor Hani, I didn't use it at all, but I know it from the literature. And when I first read about almotuzumab, I actually hated, because it resulted in 20% de novo antibody-mediated rejection, although it is a very toxic drug. It's very lymphotoxic, associated with leukopenia, severe leukopenia, anemia, thrombocytopenia for this patient. What is
and you mentioned also fungal infections. Now.
Yes, nowadays it was mentioned that it have increased fungal infections. This is very recent. Although it resulted in reduction in they use it with only Prograf or tacrolimus. So it was promising to reduce the maintenance chemotherapy, but immunotherapy. But as you mentioned, Professor Hani, it is a very dangerous drug. Should be used very carefully. I don't encourage it. I agree with you completely. But it is present in the armament nowadays. It's still present. Although I know that from the company that produce it, they never sell it. They give it to institutions. You cannot find it in the market. It's not even in abroad. It's only institutional use. And I know from Professor Adel Bakr, he told me before, it's not even used in Mansoura since long time. They tried it, of course, but they don't agree on using it anymore. So I totally agree with Professor Hani. It's a very dangerous drug. And I think rituximab ATG rituximab or anti-thymoglobulin will be a very promising protocol, and it's not expensive.
Okay, a second question about the anti-metabolites. You know that in our COVID era now, we usually sacrifice the first drug to be sacrificed will be the MPA in already transplanted patients since a long time. We decrease the dose, or we may stop even if the patient is at risk of COVID. So do you think that and those patients, many of them are stable without a rise of in creatinine in spite of doing that? Do you think that for this step of minimization of the anti-metabolites, the MPA, we need to measure the level of MPA, or we do it just empirically like this without having a level between 40 and 60 or between 30 and 60 empirically like what we are doing now, or it's better before we decrease, we should measure the level of MPA?
I think it's better to stop it completely, especially if the patient is proved COVID, and especially if he has a pulmonary infiltrate, because this is what they do abroad. We don't have a study with good two-arm and patient who is still using it or not. We will not get this studies at all. But yes. I think we have to stop it. And what I do in my patient, I increase the dose of steroids instead of MPA, and I will stop it completely. I think you agree with me, Professor.
Yes, I agree with you. Dr. May, do you have any questions for the...
Yes, you mentioned that induction therapy has an effect on prolongation of the drug survival. So do you recommend to give all patients induction therapy going to transplantation?
Actually, in my centers, we usually, I'm working in two centers, one with Professor Ashraf Dunya in Ma'had Nasser and one in Shams under my supervision. We give all patients either small dose ETG, anti-thymoglobulin, or full dose, according to the risk stratification. We stopped using basiliximab since long time, and we give the minimal dose we can use. But you know, worldwide, 80% of patients are getting induction therapy. But still,
because our patients are all live patients.
Okay.
I know, but we don't know the epitopes or the aplets, and still, if we by 6 matches, we are afraid of antibody metal rejection. And the safety of small dose was very safe nowadays. We don't see any more PTLDs like we used to see before.
Dr.
Adil will comment.
Yes. احنا عندنا دراسة لغاية 11 سنة على السيميلكت induction، وفي دراسة أخرى عالمية على 20 years experience. ملاحظين واحد إن الجرافت والبيشنت سرفايفر did not change with induction therapy، ولكن incidence of acute rejection, incidence of severe rejection, incidence of repeated rejections, incidence of rejection necessitated antibody for treatment is less with induction.
And what do you use for induction, Professor? What do you use for induction?
Induction mainly احنا are using semi-lect، ماشي، الا اذا كان في ريسك كاتيجوري، واحنا بنعرف الريسك كاتيجوري عندنا اللي هي ليه ترانسبلانتيشن او loss of the first transplant due to antibody mediated rejection.
Thank you.
Professor Adil, I have a question also for you.
Yeah.
You know that in general, the transplant tourism is, of course, is condemned by many authorities and by Istanbul Declaration and by the TTS, of course. But what is your opinion about
yes.
what they are doing in the Eurotransplant program when they can't because it is the United Europe, so they transfer the kidneys from one country to another. It's a rather a political group, not the same country. It is, again, it's a sort of tourism. What about that? Eurotransplant.
Okay.
Why they are not calling it
in Saudi Arabia and Kuwait, where yes, Saudi Arabia, they imported marginal kidneys and transplanted them around was 1992. 40 cases within the same, successively. And Georgia Buna, when he was in Kuwait before Iraq invasion, collect the marginal donors and the as Professor Ghunem asked me to assess the Arab transplant to distribute organs irrespective to the first nationality of each one, to be a global agreement, knowing that it is Europe, not antibodies.
Yeah.
So,
okay. Yes. دكتور عادل
نعم.
اوكي كمل صوتك واضح كده اتفضل كمل.
ايوه ف to do this
صوت مش واضح شويه. اوكي. يعني for the sake of time, thank you very very much for the speakers, Professor Adil Bakr, Professor Magdi Sharqawi, and for the co-chairs, Professor May Hasaballah, and also Professor Adil Bakr.
Group, not transplant.
Okay. And I think we will continue to move to the next part of our symposium today. Because we still have a very interesting speakers from Sandoz, and we will start by Dr. Peter Effat, head of the medical affairs in Sandoz, Egypt, who will give us a briefing about what is coming in the near future. Dr. Peter.
اوكي، مساء الخير على حضراتكم جميعا. في البدايه طبعا بشكر سعادتك دكتور هاني على الانترودكشن، يعني فور يور كايند انترودكشن. بشكر كل الكوكبه من اساتذتنا الكبار in reaching our very special events. يمكن النهارده احنا قصاد اول مره يكون في فيرتشوال لونش، احنا طبعا في اكسبشنال تايم. ان شاء الله يا رب ربنا يعدي الازمه دي بسرعه، وان شاء الله نشوف حضراتكم كلكم فيس تو فيس باذن ربنا، ونتقابل تاني، نرجع تاني فيس تو فيس. طبعا بشكر حضراتكم جميعا، بشكر كل المشاركين النهارده، بشكر الكوكبه من الاساتذه من اساتذتنا اللي مشاركينا النهارده في اور لونش ايفنت، وبشكر طبعا الجمعيه المصريه للنفرولوجي والترانسبلانتيشن فور دوينج ذا هول ايفنت اندر الامبريلا بتاعتها. اسمحوا لي ان فيو مينتس انا هجو ثرو سريعا، هدي اوفر فيو على شركه ساندوز. يمكن شركه ساندوز هي واحده من الاندستري ليدنج ديفيجنز من 3 ديفيجنز، هم دول اللي بيكونوا سوري انا اسف، اللي بيكونوا شركه نوفارتس. يمكن شركه نوفارتس العالميه هي 3 ديفيجنز: نوفارتس اونكولوجي، نوفارتس فارماسيوتيكالز، دول المسؤولين عن الجزء بتاع الانوفاتيف ميديسنز، والديفيجن بتاع ساندوز هو الجزء اللي مسؤول او الديفيجن اللي مسؤول عن البايوسيميلرز والاوف بيتنت ميديسنز. About Sandoz، يمكن شركه ساندوز is operating globally في اكثر من 160 دوله across the whole globe. We are leader في 3 therapy area and ranked as number 1 globally. First one which is الاريا بتاعت البيولوجيكس او specifically البايوسيميلرز ب3 marketed products with very promising and very strong future pipeline باذن الله جاي في forthcoming years. تاني جزئيه هي الجزئيه بتاعت الانتي انفكتيفز. Sandoz is global leader in the supply of antibiotics worldwide. ويمكن احنا تاريخنا في الانتي انفكتيفز بيرجع لبدايه الخمسينات where we discovered and we provided اول اورال بنسلين. ثالث واحده او ثالث ثيرابي اريا Sandoz is leader في worldwide هي الجينيريك اونكولوجي ببورتفوليو exceeding ال 50 product across different treatment modalities starting from كيموثيرابي، الهرمونال ثيرابي، السبورتيف كير، ووصولا للبيولوجيكس. اكشولي هو البورتفوليو بتاع شركه ساندوز اتس فيري برود وان. احنا عندنا اكتر من 1000 مولكيول مور اور ليس اوبريتنج في كل الميجر ثيرابي ارياز اسايد فروم اللي انا قلتهم قبل كده، البايوسيميلرز والانتي انفكتيفز والاونكولوجي. احنا عندنا كمان وي ار اوبريتنج في ريسبيراتوري، سي في اس، سي ان اس، بين، واخيرا اللي هو البورتفوليو اللي احنا النهارده بنسعد ان احنا بنعمل له اللونش اللي هو الترانسبلانتيشن اند اميونو سبريسيف ثيرابي اللي هو الرينج الكامل من التاك ومن المايكروفينولات. اكشولي احنا من 3 سنين احنا قدرنا نوصل لحوالي اكثر من 500 مليون بيشنت وورلد وايد، and our aspiration in very few years ان احنا نوصل ل 1 بليون بيشنتس نقدر نفيدهم بالادويه بتاعتنا. عايز اقول زي ما حضراتكم عارفين انه it goes without saying وده ما فيهوش اي ديبيت ان المودرن ميديسنز has changed the world، يعني غيرت فعلا في العالم، لكن unfortunately ما حلتش مشكله الاكسس. يمكن ال 100 سنه اللي فاتوا حصل هيوج ميديكال بريك ثرو، سواء ستارتنج من الديسكفري بتاع البنسلين ووصولا للكاتنج ايدج كانسر ميديكيشنز، لكن ستيل احنا عندنا النهارده اكتر من 2 بليون بيشنتس is lacking access للميديكيشنز they need. ده including كمان even الديفلوبد كانتريز مش بس الديفلوبينج كانتريز. برضو الانكريزنج هيلث كير ديماند، يعني برضو زي ما حضراتكم عارفين it has been estimated انه في خلال ال10 سنين الجايين هيكون التوتال population globally هيوصلوا لاراوند 8.5 بليون population plus الرابيدلي aging population it has been estimated انه في بعد 10 سنين هيكون في حوالي اكتر من 1.4 بليون دول بيبقى سنهم معدي ال 60 سنه، مع جلوبال هيلث اكسبنديتشرز استيميتد ان هو هيوصل في خلال 5 سنين ان هو يوصل لحوالي 13 تريليون يو اس دولار. ده بيحط increasing pressure على الهيلث كير سيستم. That's why احنا بنقول انه الجينيريك ميديسينز are key for the sustainability of the healthcare systems. يمكن على سبيل المثال في خلال سنه 2005 ل 2015 الاوف بيتنت ميديسين سمحت انه دبل النمبر، مرتين عدد الاوروبيان بيشنتس قدر يبقى ليهم اكسس للكي ميديكيشنز they deserve at the same budget. And finally اخر سلايد عايز اقولها لحضراتكم، احنا our purpose is to pioneer access to patients. يمكن هو ده البيوتي او هو ده يعني الدرايفر الاساسي لينا، ان احنا نكومباين او نقدم فورمولا which is combining the multinational standards and the highest quality medication at the same time at affordable prices تقدر تقدم access، نقدر نوصل لشريحه اعمق واكبر من المرضى. We work closely مع scientific partners بتوعنا to make access happen and we work closely عشان نقلل البردن اللي حاصل على الهيلث كير سيستم، عشان نقدر نقدم شويه سبيرنج للهيلث كير سيستمز بادجت، عشان ندي كمان مور سبيس للاذر اسينشال ميديكيشنز. ثانك يو سو ماتش اجين لكل اساتذتنا ولكل المشاركين في اللونش ايفنت النهارده بتاع ترانسبلانتيشن. ثانك يو سو ماتش دكتور هاني.
Thank you very much, Professor Dr. Rita Allouay will be the next speaker. Thank you, Dr. Peter, very much. And Professor Rita Allouay is the director of the transplant clinical research and professor of medicine in University of Cincinnati. And of course, thank you very much. She will enlighten us with her talk, and we hope to see her face to face after the COVID era. Professor Rita, please. It's our pleasure to have with us today Professor Rita Allouay. She is a research professor of internal medicine and director of the transplant clinical research in Cincinnati, Ohio. And she will be enlightening us about the target now of having better access for patients, for more patients by the new era of generics. Please, Professor Rita.
Thank you very much for the honor to present to you today. I would just like to make a personal comment. I don't know if you're familiar with them, but Dr. Osama Gaber, who is the president-elect of the American Society of Transplant Surgeons, is from Cairo, Egypt, and yes, he was my mentor. Well. Yeah, he's my mentor. He is the reason I am here today, and I am most grateful for him. So I wanted to share that because he is very, very special to me. So the focus of today's talk is generics, good medicine, or cheap fix. And I'm going to focus on the U.S. experience in kidney transplant recipients. I have the following disclosures to address. I'm on the advisory board for Veloxis and Sanofi and a speakers bureau for Sanofi and Veloxis. I've been provided an honoraria for this talk. My talk does not include any off-label or investigational use agents. The objectives of this talk today are to identify the pharmacokinetic parameters which are evaluated to determine bioequivalence in healthy volunteers, and also review the current bioequivalence data with generic immunosuppressants in kidney transplant recipients, and identify the impact of the adoption of generic tacrolimus on U.S. transplant recipients in our experience. So I just would like to start with the basic definition of bioequivalence. The regulatory definition is what's listed here. And basically, it's the absence of a significant difference in the rate and extent to which the active ingredient is available to the site of action of the drug. So the debate, you have to be willing to accept if you can get tacrolimus in the blood at the same rate and extent without a significant difference, you will have the same biologic effect. So that is the basis and the premise for bioequivalence testing. The historical perspective of this is that in the US, the FDA has had this approval process in place for over 30 years. The process is still heavily debated and is fueled when generic equivalents for cornerstone treatments such as tacrolimus are introduced in a new therapeutic area. The debate is really fueled by a lack of understanding of the generic approval process. And also what happens is when these debates occur, the FDA defends their standards and the brand manufacturers attempt to discredit the process. In the US, we had a development in 2012 where under the Generic Users Drug Fee Act, there were additional resources provided for studies to be done with generic products in the target patient population. And I'm going to be presenting that to you today, of which I was the principal investigator. So generic products must be the same in terms of active ingredients, okay? So while there may be several formulations of tacrolimus, there's a rather limited supply of tacrolimus as an active ingredient, which is used by a variety of manufacturers to develop their dosage form. Generic products must have the same dosage form. So for example, if it's capsules, it has to be capsules. If it's tablets, it's tablets. The route of the administration has to be the same. The strength and concentration has to be the same. So with tacrolimus, we could only have a 0.5, 1, or 5 milligram strength of tacrolimus. We couldn't have a 2.5 or a 3 or anything such as that. However, these products may be different in terms of shape, scoring, release mechanisms, excipients, packaging, and expiration time. Now, this is where the debate begins. Standard design bioequivalent studies are just performed as a two-way crossover in 24 to 36 healthy adults when they're only administered one single dose of the generic product and the brand product, and then pharmacokinetic parameters are calculated, and these assessments are conducted. Now, as we talk about this, you can see here on the diagram what happens. The healthy volunteers are randomized to receive a dose of brand first, so they would receive the exact same dose of the brand name product, and then a pharmacokinetic time curve would be captured by sampling the blood at various time points, very concentrated along this curve to create this curve. At the same time, another group of patients would receive the same single dose of generic with this time samples collected, the blood concentration time curve conducted. After a significant washout period, they would be crossed over and would receive the second dose of the alternative product. Based upon these resulting time concentration curves, we will compare the mean AUC, which represents the extent of which the patients are exposed to tacrolimus, and also evaluate the mean concentration max or peak that would evaluate the rate of exposure. So us in transplantation are very familiar with pharmacokinetics, I have to say. The concentration max or the peak occurs at the highest concentration by visual inspection. And the same is a visual inspection comparison of when the time to maximum concentration occurs. But most importantly is the evaluation of the area under the curve, or the AUC. And essentially, it's just a calculation of the graphic area that's under this curve to evaluate what the exposure is to the patient, or if you refer back to the regulatory definition, the extent of the exposure to the generic product. Now, FDA bioequivalent standards currently say that a one-sided test procedure with a 90% confidence interval is acceptable. This recognizes that there will be a difference in the mean values between treatments, but these differences will assume that there's no reasonable assurance that the treatment differences will be different and the treatment differences will remain acceptable. In the US, the standard procedure guidance looks at confidence intervals of 80 to 125% for the AUC. However, the EMA guidance for narrow therapeutic index drugs such as tacrolimus chooses to narrow the AUC for narrow therapeutic index drugs to 90 to 111%, and as a way to address any additional concerns that may exist for a tacrolimus product. And then also they evaluate the Cmax and make sure that the confidence interval falls between 80 to 125%. So the first generic that was available in the United States was the Sandoz generic. We conducted this study in 68 kidney transplant patients. The kidney transplant patients were evaluated 14 days prior to the study and were identified as stable patients free of rejection. They were randomized at the time that they presented for the study in a 1-to-1 fashion, and then they fell within 2 sequences of treatment. So the kidney transplant recipients received their exact same tacrolimus daily dose for the first 14 days, and then after 14 days, they were converted to the same milligram of generic tacrolimus, and vice versa with this sequence. However, during this study period, there were 4 12-hour pharmacokinetic profiles that were conducted, one on day 7, day 14, day 21, and day 28. And as you could see, that would allow us 2 12-hour pharmacokinetic parameters to assess with each of the products in all of the patients. So when you look at the tacrolimus time concentration curves that we're used to seeing with time on the x-axis and concentration on the y, you'll see here in the solid line the reference tacrolimus branded product here, and in the dashed line, the just Sandoz generic product in the dashed line with the corresponding standard area of the mean bars here. And you can see that I have widened this graph as much as I could so that you can see the differences that do exist, but they are not statistically different. When we calculated the pharmacokinetic parameters that are important for bioequivalence testing, you can see we calculated the AUC and the Cmax. And when we compared the ratios of the brand to generic, you can see that there was only a 2% difference, whereas the generic was 2% higher in AUC than was the brand. The 90% confidence intervals were met, and the p-value was not statistically significant. When we look at Cmax, the brand product was only, on average, 9% higher in Cmax, and it fell within these confidence interval. Now, to avoid confusion, I want to say this is not the bioequivalence testing interval here. This is just a comparison statistically of these pharmacokinetic parameters. So basically, the Sandoz generic tacrolimus, or Adport, was bioequivalent to Prograf in kidney transplant patients. Now, with this, there was significant public concern that was expressed in the United States and really has been expressed in several countries with the introduction of tacrolimus as an immunosuppressant, since we rely on it so frequently for the success of our transplant recipients. Obviously, the last thing that any of us who spends our life advancing the field of transplant wants to do is hurt a transplanted organ. So basically, the public concern stemmed from the thoughts that standard bioequivalence approval process based on these two-way crossover studies I've described in healthy individuals were not a valid approach for transplant patients. And they cited that there was a lack of data in transplant patients, especially high-risk patients, and referred to them as being poor absorbers, reflecting the genetic differences that occur in the liver and the gut that impact transplant pharmacokinetic parameters. There was also a need to recognize that tacrolimus was a narrow therapeutic index drug, thus, therefore, more strict bioequivalence standards should be observed, and that there were a lack of comparison of bioequivalence of generics among patients. So as I'd like to point out, the healthy volunteer studies always tested one generic product against the brand, but there were no requirements to test multiple generics against each other. So there were several consensus documents that were published at the time. I want to know, just reflecting the US and the European data, note these manuscripts date back to 2003, 2011, 2009, 2012. So quite a bit of time has passed since these publications. Now, this is a misconcept that I want to address. People who vilify or try to state that bioequivalence testing for generics is invalid, I want to remind everyone that bioequivalence testing is not only just for generics. Bioequivalence testing is used throughout the pharmaceutical industry anytime any new formulations, new dosage forms, new milligram strengths, changes in manufacturing processes are done. Bioequivalent studies in healthy volunteers are what are implemented to show that these products remain the same and provide the same exposure to the patient. I'll give you just an example within the transplant literature. As all of us know, the Sandoz oral solution was what was used in the initial phase 3 studies, but through bioequivalent studies, Sandoz capsules were developed. In the phase 3 studies for Neoral, those were oral solution. Bioequivalent studies gave us Neoral capsules. The same thing has happened for mycophenolate and tacrolimus. Tacrolimus was initially tested in 1 milligram capsules, but then underwent bioequivalence testing for 5 milligram capsules. Now, an interesting caveat, as we know, is the Rapamune oral solution for the phase 3 study was developed in a 1 milligram per ml solution. However, when they attempted to develop a 1 milligram solid dosage form, they could not come up with a bioequivalent product. Therefore, to get the Rapamune on the market, they had to repeat some of their phase 3 studies with the oral dose, with the capsule or the tab dosage form. So bioequivalence testing is not just for generics. So to give you a history of immunosuppressants, generic immunosuppressants in the United States, in 1998, the first modified release cyclosporin liquid was introduced to the market. It was later withdrawn from the market because it was found not to be bioequivalent in apple juice. However, it had been bioequivalent in several other liquid formats prior to that. In 1999, the first modified cyclosporin tablet was marketed. In 2009, actually prior to tacrolimus, the first mycophenolate mofetil generic was marketed. And following a few months later, the first generic tacrolimus was marketed. And I want to point out these times for you because what essentially happened is in the US, our two major immunosuppressants went generic at virtually the same time. And I'm going to show you in some later slides the market uptake of that existed. And whether you use steroids or not, as we all know, prednisone is all we get of that is generic. In 2013, which was only a few years later, there was already significant market penetration of greater than 50%. And I'm going to show you more granular data on that in a second. And in 2014, the first sirolimus generic was marketed. And now everolimus is lost patent, but I do not know, for example, right now if there is an everolimus generic, but I will check. So back to the funding that the FDA were provided to do additional studies, they released an RFA, and the RFA's purpose and goal was to compare the pharmacokinetics of, as you can see, 2 sources of generic tacrolimus to Prograf in stable transplant patients. And they wanted to see that patients would be switched between brand and generic to brand. So a variety of combinations. And they wanted the outcomes of the study to address the public concerns related to generic tacrolimus. So in response to that FDA and RFA, we submitted a protocol, and it was awarded to us in 2012. After that, we had a meeting assembled with the FDA, and in an effort to provide scientific input on the design from a variety of people within the transplant community. So you can see representatives of the FDA, American Society of Transplant, American Society of Transplant Surgeons were there when we, the grant awardees, presented the protocol for review, and there was unanimous consensus of the protocol design as submitted to proceed with being conducted. And the design was a fully replicated, blinded, 6-period crossover study in 2 centers. The population was essentially just stable kidney transplant patients receiving twice daily tacrolimus who were not on any interacting drugs. Our intervention was to first identify of the 5 to 6 generic tacrolimuses that were currently available on the market, just looking at the data that we had, which was the healthy volunteer data, could we potentially predict what would result in the highest concentration of the generic and what would result in the lowest concentration. And from that, conduct 6 pharmacokinetic assessments over a 12-hour period, twice with the brand, twice with the generic high, and twice with the generic low. So again, this study was conducted in 36 kidney transplant patients. We measured not only tacrolimus, but its metabolites in LC-MS, and we did genetic phenotyping of all the subjects. So patients were screened and randomized on day 1 to fall into one of the 3 treatment sequences. And so you can see, as I described before, it's a variety of treatment sequence that were generated by the computer that randomly assigned them to the various 3 products, but also administered the brand twice, generic high twice, and generic low twice. Now, that's very, very important point that will come up later, because when we have these 2 measurements in the same patients, we're able to compare the within subject variability, which is very important. And after each of these were administered, again, a 12-hour PK was conducted at 6 different time points. Now, blinding method of this study, we initially discussed over-encapsulation of the product, but there were some concerns that were related that may interfere with the pharmacokinetic properties. And so our blinding methods continue to be, we at the pharmacokinetic site were blinded as to what sequence the patients would be enrolled to, but we could see the drug that we were dispensing to them in our hands. However, when we collected the samples and sent them to the analytical site for measurement of the levels by LC-MS, they did not know which treatment group the patients were in. And once those level results were sent to our pharmacokinetic analysis site, they did not know what the patients were receiving as well. And it was not until after all of the pharmacokinetic parameters had been calculated that the investigational drug pharmacy unblinded the pharmacokinetic team to the products. And so for a little bit of fun, I'm going to keep you guys blinded to the formulation until the conclusion of the presentation. So again, at this time, there were 5 generic tacrolimus products that were available. We identified the 2 most disparate. The one that was predicted to be generic high based upon the criteria that we had was very clearly identifiable based upon our selection criteria. However, the product that we predicted for generic low, there were 2 products that were very close in the standards. However, candidate 1 did not have enough of the formulation on the market for us to get the same lot number of the product. And obviously, we had to have the same lot number. So we ended up going with generic low candidate 2. And as well, with any pharmacokinetic study, as you know, adherence monitoring is very, very important during the trial. We had weekly assessments when we saw the patients of their adherence. We dispensed the medications in the bottles that had a computer chip in the top of the bottle that would tell us when they open and close the bottle. Obviously, it didn't tell us if they actually put the medication in their mouth, but we could see when they open and close the bottle, and you could see that we had very good sampling of adherence from that perspective. We also did a patient diary review and pill counts. And in addition to that, I'm not going to present the data today, but a finger stick was conducted daily on all of these patients, and we were able to, via the finger stick technology, assess what the tacrolimus levels were every single day of the patients when they were at home. Now, per protocol, patients were excluded from the PK analysis if they missed a dose within 48 hours, as assessed by any of these mechanisms, or if we did not have all 6 of the pharmacokinetic parameters to evaluate for the final analysis. So this is a busy slide, but I'll just say, obviously, we had to evaluate a lot of patients to find patients who were eligible, who could, meet the criteria and also be able to undergo such a rigorous study. We randomized 41. Of the 41 that we randomized, 1 didn't show up for their baseline visit. After we had the 40 enrolled, 1 patient was withdrew because of elevated liver function test due to a nutritional supplement. 1 developed a UTI, and 1 was unable to maintain venous access after the first PK. When it came time to analyze the results, we analyzed 35. We had to exclude 2 because there was a dosing error on the last PK visit in 1 patient, and then there was a non-adherence within 48 hours of 1 of the PK visits. So in terms of demographics, it's a typical a U.S. population with a mean age of 52, 65% males, approximately 20% African Americans. The transplant donor type was reflective of our center. The time post-transplant was roughly 4.5 years. Only 14% of the patients were on steroids, which reflects our current standards, 100% CellCept, and the mean daily dose of tacrolimus was 5 milligrams a day. So I'm going to take a second to orient you to this slide because I'm going to go through several of these really quickly. Again, this is the pharmacokinetic time concentration curve with time and concentrations along this axis. And these open dots represent every time that we collected a tacrolimus blood sample, and these error bars represent the variability between the patients, okay? This is within patient variability. So this is when we gave product 1 the first time we gave it. This is product 2 the second time we gave the product, and you can see how they match up here. And again, note that this is a relatively wide scale here. This is when we gave the second product the first time. This is when we gave the second product the second time. The third product the first time. And the third product the second time. And when you look at all of these curves superimposed on top of one another, there's no statistically significant difference at any time point of these curves. However, just visually looking at the curves is not the most optimal way to evaluate pharmacokinetic data. It requires us to look at the pharmacokinetic parameters. And so, as you can see here, we looked at the AUC, Cmax, Cmin, and Tmax. And the observed AUC for the brand product was 92, generic high 97, generic low 98. The Cmax 14.4, 15.6, 15.8, and so on. And again, these are the Cmins, which would represent the trough in these populations. And again, keep in mind that this is a 12-hour PK on the AM morning dose, and the Cmins or the troughs maybe not exactly the same as we look at a trough from a morning dose. Now, we went one step further to do the most rigid analysis of this data as we could, and it was called scaled average bioequivalence. And I won't belabor it too much, except to say that drugs with a more narrow within subject variability could have the potential of having a clinically significant difference if they were widely variable. So what scaled average bioequivalence does is if you're within subject variability is right around 20%, 22%, the range of 80 to 125% to meet bioequivalent standards remains the same. However, if you're within subject variability is less than 20%, these limits become more narrow or make it more rigid for the drugs to be approved. And just as a point of reference, it's not until within subject variability of drugs is greater than 30% that from an FDA perspective, they're considered to be highly variable. So as we move through this, I'm going to show you the tacrolimus assessment of AUC by scaled average bioequivalence in the kidney population. You can see when we compared product 1 to product 2, with product 1 being the reference, there was only a 12% within subject variability. Product 1, again, 12% variability. When we compared product 2 to itself, 14% variability. Product 3 to itself, 15% variability. So the obvious question for this would be, from some person is, I don't believe that data. We have all written papers saying that tacrolimus is a highly tacrolimus has a high within subject variability. Why are these numbers you're showing me so low? I have even written in papers that tacrolimus has a high within subject variability. The within subject variability that we see, though, outside of controlled environments is primarily related to timing, food, and a variety of different things. When you measure it in a controlled environment, the variability is relatively low. So from that, from this variability, we calculate the scaled average bioequivalency, which is again, a limited range. You can see that these ranges that we calculated here, because the within subject variability was low, these are all less than 85 to 125%. When we looked at what we observed as the 90% confidence interval, which would be what was required by the FDA, you can see that these 90% observed intervals fall within the scaled average bioequivalence limit. Therefore, it shows that these generic products passed not only the standard FDA criteria, but the more rigid scaled average bioequivalence criteria. And this is looking at AUC. I'm not going to go through it again, but essentially we observe the exact same thing from Cmax. You can see again that the reference within subject variability is higher related to Cmax. And you can observe it here, but still for Cmax, it passed all of these criteria. And then when you look at the concentration min, you can see that it passed these criteria. When you look at the adverse events by formulation, there were no acute rejections that occur, which you would expect that in a stable patient population with 100% patient and graft survival. One patient, as I referred to previously, developed pyelonephritis. However, as we know with all of our transplant patients, 71% of them reported an adverse event. But when we grouped our adverse events by the primary categories that we typically see, there really were no difference between products, except maybe if you look here at respiratory events in product 2. But I think, so I let you evaluate that data as you see fit. But in our perspective, there were no differences in adverse events. Now, it's important for us to appreciate the complexity of tacrolimus and its metabolism with its impact on clinical care, because as you know, the metabolites of tacrolimus do have immunosuppressant activity. So here, what happens with tacrolimus, as we all know, you administer Adport, it goes in through the intestine where it is taken up within the GI lumen, and then it goes through a PGP or a transport mechanism into the gut wall, which actually houses cytochrome P450 enzymes. So a lot of the tacrolimus is metabolized within this gut wall before it even makes it to the portal vein. And when it goes to the portal vein, the drug immediately goes to the liver, of which is the highest concentration of the CYP3A, when it's metabolized further and then excreted through the bile. So I think that we all know that these sources of variability are some of the problems that we see, especially with drug interactions, etc. However, when we look at the primary metabolite, 13-O-desmethyl, that is the highest concentration and the highest immunosuppressant activity, you can see that between brand, generic high, and generic low, where we looked at the AUC 0 to 12, or the ratio of these numbers, there were no difference between the groups. And when we compared this by the testing that we looked at before, all of these fell within the scaled average bioequivalence and the observed bioequivalence parameters, even for the active metabolite, which, to be quite honest with you, we weren't expecting to find this. So in conclusion of this study, with substantial input, we found that there were no difference in AUC, Cmax, or trough. There was comparable within subject variability for all formulations. There were no observed changes in organ function with similar adverse event profiles, which I didn't show you that, but there was no difference. And the covariants that we evaluated were no different between the groups. So to now unblind you, when we look at the product selection, we had, again, product 1 is brand, product 2 is generic high, product 3 is generic low. Prograf was obviously the brand, generic high was Adport or the Sandoz generic, and generic low was Dr. Reddy's formulation. And the observed pharmacokinetic parameters, there were no statistically significant differences that were observed for any of the pharmacoparameters by formulation in the transplant recipients we could observe. So to briefly move on, what is the purpose of a generic tacrolimus formulation if they're really similar to brand? And it's obviously a cost savings effect, but who actually benefits from the savings if the patient is absorbing any risk that happens to be? So the experiment that you're starting in Egypt now is similar to the experience we started in the United States back in 2009. And this graph goes from 2009 to 2014 and looks at the percent of patients who were receiving tacrolimus brand in the solid line and tacrolimus generic in the dashed line. Now, these dashed vertical lines represent every time a new generic was introduced, tacrolimus generic was introduced to the marketplace. So what you can observe here is with Sandoz generic being the first introduced to the U.S. market, there was already within a year or less than a year, a 50% switch to generic product that continued to rise until 2014. This phenomenon was observed similar with mycophenolate mofetil. The only difference in the mycophenolate mofetil situation was that there were several generics that were approved by the FDA prior to their ability to be marketed. And when they were marketed, you can see that the time period of when it crossed 50% is a lot earlier with mycophenolate mofetil than it was with tacrolimus. However, since that time, the percentage of mycophenolate mofetil of generic in the transplant population has continued to increase. This just looks at the percentage of patients that were dispensed a generic product. This is the middle line is the median, and this is the 90th percentile and the 10th percentile for both kidney tacrolimus and MMF. Now, when you evaluate the cost of transplant immunosuppressive agents after the introduction of generics, there's a variety of different things that the marketplace can do. But in the case of tacrolimus and CellSup, I can define what happened in the U.S. population by the evaluation of SRTR data in transplant recipients that were covered under Medicare Part D. And again, if you look at the reduction in drug costs from 2008 to 2013 in patients on Part D, there was a 50% decrease in tacrolimus that resulted in an approximately $1,700 to $2,700 savings per patient. With CellSup, there was a drop, a decrease in Medicare payments of 55 to 67%, which resulted in a $2,500 to $4,200 cost savings. When you look at the patients that were unsubsidized or had to pay out of pocket, those were the ones that had the greatest reduction in their costs, with tacrolimus of 61 to 72%, or $900 to $1,300 per patient, and CellCept decreased from 71 to 79% with $1,000 to $1,800. So annualized over these years from 2008 to 2013, you can see the cost decrease that was observed in the Medicare Part D payments for both tacrolimus and CellCept. And when you observe these, again, the cost savings were per patient were roughly $2,000, $2,500 for each agent per patient. So in conclusion, AdPort is bioequivalent to brand tacrolimus in healthy volunteers. The data was not presented. AdPort, but that's the data that's presented to the regulatory authorities to receive market approval. AdPort is bioequivalent in stable kidney transplant recipients with no observed changes in organ function or adverse event profile. Over 50% of the kidney population in the U.S. has received tacrolimus generic since 2011, and the introduction of generic immunosuppressant has lowered the cost of immunosuppressive agents to the provider. So that concludes my presentation.
Thank you very much, Professor Rita, for a very enlightening presentation. And also thank you for mentioning Professor Osama Gaber, who he with his wife, Dr. Liliane Gaber, came to Egypt many times, of course, their country. And we are very happy that you were a colleague to Professor Osama. Thank you very much.
Thank you.
Okay, let me introduce Dr. Amit Kumar, who is the Global Medical Manager of the Transplant Sandoz Group, to speak about this subject. Dr. Amit.
Yeah, hello, good evening. Good evening, everybody. My name is Amit Kumar Rai, and I am the Global Medical Manager for anti-infectives, antivirals, and transplant. And I am based out of Kundl, Austria. Kundl is a very small village in Austria, but it has a very special significance in Sandoz because penicillin was first produced commercially in Kundl and it is still being produced. Okay, today we are not going to talk about penicillin, but of course, today I will present you the executive summary of mycophenolate mofetil and its use in kidney transplant. So before we actually go into the details, let me just show you the agenda of today's presentation. So MMF introduction, I will introduce MMF, its mechanism of action, dosing, its approval, and everything. Then what does what important role does MMF plays in the transplant? The next we will see the positioning of MF in the global kidney guidelines, followed by which I will just explain in nutshell the clinical efficacy studies which led to the approval of MMF. Finally, there will be some discussions regarding the BE study for the mycophenolate mofetil versus CellCept. And at last, I will just highlight that MMF is equivalent to the enteric coated formulation of mycophenolate sodium. So without wasting any further time, let me start the presentation. So MMF stands for mycophenolate mofetil. It's a prodrug of mycophenolic acid and is developed to improve the bioavailability of the acid form. And mycophenolate is marketed in two different formulations. One is the prodrug mycophenolate mofetil and another is the enteric coated mycophenolate sodium tablets. So the figure which you are seeing is the ADME of the mycophenolate mofetil. Once absorbed, it is hydrolyzed by esterases in the gut wall, blood, liver, and tissues to the active form mycophenolic acid. And after that, it is metabolized in liver, GI tract, and kidney by UGT enzyme to MPAG, which stands for mycophenolic acid glucuronide. This is the metabolite of the MPA, and then it is excreted into urine via active tubular secretion. But there is another step happening, which is the enterohepatic circulation of MPAG to MPA during 6 to 12 hours after the dosing. and it almost covers 40% of the AUC of the MPA. So coming to the mechanism of action and PK/PD properties of MMF. It's a potent, selective, and reversible non-competitive inhibitor of inosine monophosphate dehydrogenase. It's and IMDPH, it's a key enzyme involved in the de novo synthesis of guanine nucleotides, leading to arrest of T and B lymphocyte proliferation. So in a nutshell, MMF inhibits the proliferation of T and B lymphocytes, and no antibodies are further produced. Hence, the immunosuppressive effect happens. After oral administration, the absolute bioavailability of MPA ranges from 81 to 94%. It is almost 99% bound to serum albumin in patients with normal renal and liver functions. It displays a linear pharmacokinetic parameter with Cmax occurring in 1 to 2 hours. And at 6 to 12 hours, I just told that a secondary peak due to enterohepatic circulation occurs, which is 40% of the AUC. The mean elimination half-life of MMF ranges from 9 to 17 hours. So coming to the approval, indication, dosing, and adverse events. MMF was approved in US in 1995 for the prophylaxis of organ rejection in kidney, heart, and liver transplant recipients. Similar approval was granted in EU in 1996. The approval was based on 3 randomized double-blind controlled phase 3 trials in de novo kidney transplant patients, which I will discuss in the coming slides in a few moments. MMF is marketed under the brand name Cellcept by Roche Pharmaceuticals, but now there are many generic versions available in the market because the patent of Cellcept has expired. Coming to the dosing, in adult renal transplant, 1 gram twice daily. In adult hepatic transplant, 1 gram twice daily IV or 1.5 gram twice daily orally. In cardiac transplant recipient, 1.5 gram twice daily orally or IV is given. I would suggest that to have a more detailed version of the dosing, physicians must refer to the summary of product characteristics of either Cellcept, or maybe they can refer for Sandoz mycophenolate mofetil. Coming to the most relevant adverse events, occur in the GI tract. For example, nausea, vomiting, diarrhea, abdominal pain, and gastritis, which might occur in up to 45% of the patients, and are often a reason for dose reduction or discontinuation. But there is nothing to worry because these adverse events can be controlled easily by reducing the dose of MMF in patients. So, what important role does MMF play in the transplant field for in the solid organ transplantation field? After the approval of MMF by FDA, it has become the first-line drug for solid organ transplantation. An estimated 79% kidney, 48% liver, and 75% of heart transplants are now prescribed mycophenolate mofetil instead of azathioprine at the hospital discharge. And here in the graph, as you can see, from 1998 until 2009, the percentage of percentage of patients prescribed with mycophenolate at discharge increases, and simultaneously the percentage of patients prescribed azathioprine at discharge decreased. MMF is a new consistent member of many different immunosuppressive regimens, especially calcineurin inhibitors, so that their side effect can easily be managed. The popularity of mycophenolate is partly based on its reputation as a drug associated with little or at least manageable toxicity, which I have just mentioned before. Coming to the Global Kidney Guidelines, KDIGO, which stands for Kidney Disease Improving Global Outcomes. And the reason why I'm showing is that this guideline suggests the use of both tacrolimus as the first-line CNI to be used, and mycophenolate to be as the first-line anti-proliferative agent. So, which means both the tacrolimus and mycophenolate, mofetil are advised by global guidelines to be used as immunosuppressive medication for maintenance therapy, along with or without steroids. So this was all about the introduction of mycophenolate. Now let's have a quick look about the studies of mycomofetil which led to the approval. So there were three studies, three different studies which led to the approval. All the three studies are randomized, double-blinded studies, and let's go into each one by one. So the first is the US renal transplant MMF study, and it involved 499 patients randomized to mycophenolate, either 2 gram per day or 3 gram per day, compared to azathioprine 1 to 2 milligram per kilogram per day. In addition to this, all patients received cyclosporine and prednisolone along with the treatment. The primary endpoint was biopsy-proven acute rejection or treatment failure within the first 6 months of following the transplantation. Coming to the results, as you can see, the BPAR or the treatment failure occurred most frequent, for almost in 48% of the patients for azathioprine compared to almost 31% in the MMF treated group. The time to first BPAR was significantly longer for MMF group compared to the treatment group. Coming to the safety, the frequency of adverse events was similar in both the treatment groups, although GI problems, leukopenia, and opportunistic infections were more common in MMF group. So, in conclusion, this study with almost 500 patients demonstrated that MMF is highly effective new immunosuppressant for the prevention of acute rejection of renal allograft. Coming to the second study, this is the tricontinental mycophenolate study because it it involved 3 different continents, as you can see, Canada, Australia, and EU. On a similar note, 503 patients randomized to MMF and azathioprine, and similarly they received cyclosporine and prednisolone along with this. The primary endpoint was same, treatment failure of BPR within 6 months. Coming to the results, as you can see, the BPR occurred in almost 35% of azathioprine group compared to 16% or 20% in MMF group. Treatment failure occurred in 50% of the patients in azathioprine compared to almost 34 and 38% in the MMF treated group. The frequency of adverse event was similar as described in the previous study. With this study, it concluded that the benefit of MMF is evident within the first 6 months after transplantation. Coming to the last study, which is the final study, the European MMF cooperative study group. The difference between this study and the previous 2 studies is that here the patients were randomized to MMF and placebo instead of azathioprine. All patients received cyclosporine and prednisolone as in other studies, and the primary endpoint was same. Coming to the results, BPAR or treatment failure occurred less frequent in MMF group compared to placebo group. The frequency of adverse event was similar in all treatment groups. So in together, concluding all 3 trials found no significant difference in the graft loss or death at 6 months post-transplant. They all reported similar adverse events related to MMF. And finally, all these 3 results led to the regulatory approval of MMF by US and EU agencies. So as you can see, they all were limited for the first 6 months, but this EU group conducted post study for 3 years, 3-year follow-up study, and the follow-up study for 3 years demonstrated that 3-year graft survival, including death as a cause of graft loss, was better in MMF treated group compared to the placebo. And as you can see also here in the graph, the yellow bar represents the placebo and blue and orange represents the MMF treated patients. So number of patients who died in the MMF treated patients were less compared to what is in placebo. Similarly, patients experiencing graft loss and graft loss due to rejection were less in MMF treated group compared to placebo. So this is so we discussed how MMF was approved and the follow-up 3-year study. This study is just simply the comparison of MMF with azathioprine, and it shows that MMF is associated with reduction in incidence and risk of late rejection compared to azathioprine. And this was done for 6 months acute rejection and as well as 3-year rejection. As you can see in the graph, that the percentage of incidence in azathioprine treated group is more compared to mycophenolate group. So with this, I conclude the efficacy and safety studies of the mycophenolate. And now an important part, coming to the PE study of the Sandoz mycophenolate mofetil versus CellCept. As in the previous presentation, the importance of bioequivalence data was highlighted. So I will not go into detail what is bioequivalence study, because now I hope it's clear for everybody. So coming directly to the B study of Sandoz mycophenolate mofetil versus CellCept. It's a randomized open-label 2-period, 2-treatment, 2-way crossover BE study of mycophenolate mofetil from Sandoz 500 milligram tablet compared with CellCept, which is the reference product. The objective of the BE study was to compare the rate and extent of absorption of Sandoz MMF compared to CellCept. 59 healthy subjects completed the study, and 57 were included in the data set for statistical calculation. And as I will repeat again, the criteria for the bioequivalence was that the ratio, geometric mean ratio of the test and reference should fall within the 80 and 125%. So coming to the result, this is the plasma concentration time curve for both the reference and the MMF from Sandoz. As you can see, there is a perfect overlap of the graph. And here, the 90% geometric mean also, the ratio lies between the 90 to 125, clearly signifying that Sandoz mycophenolate mofetil is bioequivalent to CellCept. Coming to the safety part of the study, no serious or significant adverse events were reported, and both the formulations were well tolerated with no major side effects and no major relevant differences in the safety profiles. Hence, based on these results, it can be concluded that the test mycophenolate mofetil, which is the Sandoz MMF, is bioequivalent to the reference CellCept, following the dose of twice 500 milligram tablets under fasting conditions. So this is the slide. This is a small study just to highlight that MMF is therapeutically equivalent to enteric-coated mycophenolate, which means both the drugs can be interchanged and can be given to patients. So here, enteric-coated mycophenolate sodium is therapeutically equivalent to MMF in de novo renal transplant patients. So there are 2 slides for this. One is the efficacy comparison, and as you can see, so BPR, graft loss or death, which are the primary endpoints generally in these studies, is almost comparable in both type of formulation. Similarly, as you can see in safety comparison, any sort of adverse event were similar in both enteric-coated mycophenolate sodium as well as in mycophenolate mofetil. So these 2 slides, this study actually tells us that both the formulations are therapeutically equivalent and can be interchanged. This is just a snapshot to show that Sandoz MMF, which is bioequivalent to CellCept, has been approved by FDA also. And at last, this slide is just again to emphasize the importance of generics and why generics should be used in the transplant patients. As you can see, generic should be used because it's bioequivalent in appropriate BE studies, meets the same batch requirements for identity, strength, purity, and quality, manufactured under very strict standards, contains the same active ingredient, and is identical to strength, dosage form, and route of administration. Hence, generics should be used for the treatment of transplant patients. With this, I conclude my presentation. Thank you very much for your time, and have a good day.
Thank you very much. I think we can now have some questions from the audience. I hope Dr. Rita is with us for the. The question for Dr. Rita, do you need to follow your patient more frequently if you switch from Prograf to Adport?
With the initial experience in the United States, recommendations were made to increase monitoring around the initial conversion to a generic product. However, after the clinicians got more experience and comfort with it, they recognized that that really wasn't necessary. I think that the point that I would make is we have to recognize that every time we measure a level, if a patient is on Prograf, when they come to clinic, it's not the exact same number every single time. And what tends to happen is when they get converted to a generic, if there are any differences that are observed, it's automatically assigned to the generic product. So what I've essentially done with people that may be uncomfortable with the switch without monitoring is just kind of do a small study of your own. Switch a few patients to generic, bring them back in a week or so, test a level, and see what you think. The data shows that it's not necessary. And as you can see, the experience in the US, it has been going on now for over 10 years. And our patients are switched between generics, started on generics, switched from brand to generic without additional monitoring. However, that I will say that is a controversial statement. And some people may disagree with it, but the data does not support the need for increased monitoring, especially after 10 years experience of use in the US.
Another question.
Yes.
Do you have a difference? You see a difference between once daily tacrolimus and divided doses of tacrolimus as regards the side effects?
We have not conducted a study on that topic. However, there are several studies out there in the literature. We did do one pharmacokinetic study that actually compared twice daily Prograf, once daily Advograph, and once daily Envarsus or Veloxis to actually compare the pharmacokinetic parameters. The literature out there is that you may see some decrease in the neurologic tremors that are associated with the peak toxicities that are associated with specifically the Envarsus formulation, but I would refer you to that literature for reference.
A question for Dr. Amit, please. Okay, Dr. Amit, you hear me?
Yes,
okay.
I can hear you. Thank you.
You mentioned that the mycophenolate mofetil is equivalent to sodium the
enteric-coated formulation. Yeah.
But I think the previous reports were that sodium mycophenolate may have less GIT side effects than mycophenolate mofetil. So there is a question about that. Is it still valid, this less GIT side effects or not?
Yeah, I mean, you're absolutely right. There are many reports which we came across tells that enteric-coated formulations are better tolerable compared to mycophenolate mofetil, especially in terms of GI side effects. So you're absolutely right with that. But I still do not came across any reason like why MMF is preferred over the enteric-coated formulation.
Okay. A question for Dr. Adel Bakr.
Yes, Dr. Yes, Dr.
Adel.
Yes.
Dr. Adel, are you doing any survey about the history of transplantation, not only in the Arab area, but in Africa and in more, in the more surrounding areas, or you are not planning to do this?
Yes, I'm preparing with Mehmet Habral to do history of transplantation in Middle East countries to add to Arab and Middle East areas.
Okay, we are waiting for this because it will be also interesting to hear about the nearby countries around us.
Moreover, I tried to translate this book into Arabic through an agent in Syria, starting translation and then revision. Then I prepare the chapters to each author before publishing it in a very good manner.
Dr. Amaya, you have any questions for them?
Could I ask you where else is AdPort being used? What countries is it available in? The AdPort? Where is it used, the AdPort? What countries is it available in? And the microphone اللي مفاتيح as well.
Dr. Rita.
I thought that then it's probably best for the Novartis person to ask that question, answer that question. But the Sandoz product is available in the U.S. and has been available here, and it's the same product. And it's a branded generic apparently with AdPort for you guys.
And how long has it been used in the U.S.?
Since 2009.
Very long period, very long period. Regarding pharmacoeconomics of the use of AdPort and also of, you mentioned it is around 50% reduction in the total cost, or it could be less than that even. What? It depends. Of course, it will depend on the price it will come to Egypt. I'm not sure. In the US, it is 50% reduction?
For the answer from myself is yes, it's a 50% reduction. I think pharmaceutical companies take a different model when they reduce prices. I think if you look at CellCept, the conversion happened quicker and there was a slightly larger price benefit. Usually when generics are introduced into the market, there's a 75% price reduction. However, it's only been approximately 50% with the two primary transplant immunosuppressants.
Very good. Thank you.
Yes.
So I think we can conclude our very interesting meeting tonight for better access for patients. And thank you very much for the speakers, for Professor Adel Bakr, for Professor Magdi Sharqawi, for Professor Rita Alwar, and Professor Amit. And thank you very much for the co-chairs, Professor May Hasaballah and Professor Adel Bakr. And thank you all who waited for us for around almost 3 hours now. We started at 7 o'clock. Now we are approaching 10 o'clock according to Cairo time. It was but it's I think we got a lot of information, and we went in the history of transplantation and in the future of immunosuppression with Professor Magdi Sharqawi. And we went also with a big knowledge of the because doctors usually are not very good in all these stories of pharmacogenetics, pharmacoeconomics, but you enlightened much as you have a pharmacy base about many of these concepts. Thank you very much about the narrow therapeutic index and all these things. And also, Professor Dr. Amit, you enlightened us much about the comparison between the MPA and all the studies stressing its role in transplantation. Thank you all, and hope to see you face to face after the COVID era. Thank you.
Yeah, and also hope to see you soon tomorrow also, right, for the second day.
Yes, thank you very much.
Perfect. Thank you, everybody. Have a nice day.